Hi everyone, and welcome back to the next review video for the biology or living environments regions. This is Unit 4, Topic 1. So the first thing we're looking over is the difference between autotrophs and heterotrophs. So autotrophs, or autotrophic organisms, are organisms that can make their own food. And this is normally done through photosynthesis. So some examples of these are plants or forms of photosynthetic algae that are found in the ocean. So these really take light energy or energy from the sun, and through the process of photosynthesis, create their own glucose, which is the food they need for other processes. Whereas heterotrophs are organisms that cannot make their own food. And some examples of these are humans, like us, the form of the fungi, and in order to get their energy, they need to eat other organisms. So we are heterotrophic, we get our food, we can't make our own food from sunlight. We have to eat other animals, whether it's plants, animals, we have to eat other organisms to help us gain our energy. So heterotrophs eat others, where autotrophs make their own. So now we're going to go into plants, which are one of our main autotrophic organisms. The first thing we're going to look at are the parts of plants. So the first part of plants are the roots, and the roots absorb water from the soil. So this one is pretty straightforward. You know the roots are deep underground, and their role is to help absorb water. Next, you have the stem, which runs along the leaf, and this is transporting water. And it transports water from the roots to the leaves, where the leaves use the water. And then the final part you want to know are the leaves, and the leaves are where photosynthesis occurs. So the leaves of the plant are really where this process is going to happen. So we're going to kind of zoom into the leaves now, because this is really the most important one, especially when we're looking at photosynthesis. So the first part you want to know is the palisades layer, which is this thick layer you can see on the diagram to the right. And the palisades layer is where photosynthesis takes place. This is where the majority of photosynthesis is going to occur, and it's where you have these tightly packed cells. The next part of a cell leaf you need to know, which are seen at the bottom, are those little gaps, and those are the stomata. So the stomata are these little pores or gaps in the leaf where gas exchange occurs. So CO2 is going to enter the plant so that it can be used for photosynthesis, and oxygen that is produced is going to exit. So the stomata are the little holes in the epidermis where gas exchange occurs. And finally, you have the guard cells, which open and close the stomata. So these are really controlling the water loss and controlling this gas exchange, as when the guard cells are open, the stomata is open, and gases can go in and out, and also water can go in and out, and when they're closed, it really stops that. So it opens and closes the stomata. So now we're going to look at the process of photosynthesis. So the location, specifically the location within the cell, we know it occurs in the leaf, but within the specific cells, this occurs in the chloroplasts. So these chloroplasts are those specialized organelles that are found only in plants, and that is where it's going to occur. So now our equation that you want to remember for photosynthesis is that carbon dioxide plus water are reactants. So the plant takes in carbon dioxide and water, and then using light energy, so we'll just draw the little arrow with light above it to show that light energy is being used, it produces glucose and oxygen. This is the key one. This is really the key thing to remember about photosynthesis. It's also the opposite of cell respiration, which we'll look at in a little bit. But carbon dioxide and water go in, and glucose and oxygen go out using light. So there are various factors that affect photosynthesis. The first is light intensity. The amount of light and also things like the color of light can affect photosynthesis. As you can see on this diagram here to the right, as light intensity increases, the rate of photosynthesis increases, and then it eventually flatlines. So like around there is about the maximum level of photosynthesis. So even if you increase light intensity even more, it'll level out around there. So you want to know some of these graphs to the right. The second one that changes it is the amount of carbon dioxide. So carbon dioxide concentration in the environment is going to affect the rate of photosynthesis. And this one also has the graph for this is pretty much the exact same as the light intensity graph. And that's because if you think about it, when the cells are all operating at their maximum capacity, even if you have more carbon dioxide, there are no more, empty or unused chloroplasts to conduct photosynthesis. So it gets to a point where it's at its maximum efficiency for rate of photosynthesis, and the same occurs with light. Even if there's more light energy, all the chloroplasts are already undergoing photosynthesis, so more light doesn't change anything. And then the final one is temperature. And looking at temperature, whether it goes too high or low above the optimal, can affect the rate of photosynthesis. So on the graph here, we can see the optimal temperature is about right there down the middle, and anything that's either too high or too low will cause photosynthesis to decrease. So you want it to be as close to the optimal as possible. Okay, now the next process we're going to look at is cell respiration. And one thing I'm just going to write here at the top, because it's a common misconception, is all organisms carry out cell respiration. Oftentimes, some people think that, because we always talk about cell respiration in like humans and other animals and photosynthesis in plants, only plants do photosynthesis, but all organisms must do cell respiration. So this is something that that plants will also be doing in the mitochondria, because plants also have mitochondria and do this. So I kind of just gave away the location, but the location for cell respiration in the cell occurs in mitochondria. And these mitochondria are the specialized systems that are used to create this ATP. So the equation for cell respiration is really the opposite of photosynthesis. So you take oxygen and glucose. So oxygen plus glucose go in, and then that creates carbon dioxide, water, and then this big key one at the end is ATP. So ATP is really the most important part of cell respiration, because ATP is the energy that will be used for all other processes throughout. So the entire point of cell respiration is to create this energy to be used. And now there are the two different types of respiration. You have aerobic respiration, which is really your regular cell respiration when oxygen is present. So the key with aerobic respiration is that oxygen is present. And this creates your regular ATP yield, which is about 36 to 38 ATP, and this only occurs when oxygen is present. In contrast, there's anaerobic respiration, which is when no oxygen is present. And anaerobic respiration is really the second choice to aerobic respiration, because it creates about 2 ATPs instead of the 36 to 38 ATPs. And there are also two different types you want to know. The first is alcohol fermentation, which happens in yeast, which produces, as it sounds like, alcohol and 2 ATP. And then there's lactic acid fermentation, which occurs in animals, like in our muscles if we run out of oxygen, which produces lactic acid, as implied by the name. And the other thing to just remember about lactic acid is that it's thought that lactic acid causes our muscle fatigue during exercise. So when you're exercising and your muscles get really sore, that's from a buildup of lactic acid from anaerobic respiration. Okay, so now we'll look at some review questions. First one, we have number 29. The diagrams below represents a response that occurs in the guard cells of plants. So sun is shining and large water is available, causes guard cells to open leaf pores, and the sun is shining and little water is available, causes guard cells to close leaf pores. And we're asked, the change in the guard cells activities illustrate. So this is really a question about controlling and the opening and closing of the stomata. So first we say an immune response intended to limit water use. So this would not be our answer, because it's not like a pathogen is invading, which would trigger the immune system. It's rather just trying to to maximize the amount of water. The next one we see is a passive transport in response to the sun shining. This isn't anything about passive transport across the cell membrane. The stomata and opening and closing stomata are not changing anything about a cell membrane, but rather it's just a gap in the outer layer of the cell. So this would not be passive transport. The next one is a feedback mechanism to control water loss. This one would likely be our best answer, as if we remember from earlier units, that feedback mechanism is when you're responding to some environmental change to help maintain homeostasis, and if in this case it's controlling water loss, we see that responding to either a large amount of water available or a little amount of water available triggers these results to occur. So this would likely be a feedback mechanism. But just to be sure, we'll look at number 4. Number 4 says genetic manipulation caused by the presence or absence of water. This would not be our answer, because guard cells opening and closing is not changing anything about the genetic sequence or the DNA anywhere. It's just whether or not the cells are contracted or relaxing to open up these stomata. So number 3 would be our best answer. Next is number 18. That says, while looking at the bottom surface of a leaf with a compound light microscope, a student notices pairs of cells with openings between them on the surface of the cell. The main purpose of these openings and the cells that surround them is... So this is also another question about the stomata and the guard cells. So one is removing excess sugars, which is not the function of the stomata. Number 2 is synthesis of carbon dioxide, which does not occur there. Carbon dioxide is used for photosynthesis. Number 3 is regulating gas exchange, which we know is the answer because it controls CO2 entering the cell and O2 leaving the cell. And finally is purification of water, which just is not done at the stomata. So number 3 would be our best answer here.